everyone. Today, let's imagine ourselves as water molecules. As you can see, water molecules can freely and randomly move in a solution based on its intrinsic kinetic energy. But what will happen if water molecules are separated by a membrane? Now, the movement of the molecules is disrupted and will be determined by a concept known as water potential. So welcome to BioWorld, where we're going to discuss how water potential affects osmosis. According to our STPM syllabus, we need to understand the concept of water potential as well as another two potentials known as solute potential and pressure potential. Then we are going to do some calculations based on these three potentials. So come, join me. To help with my explanation, let me introduce to you two solutions separated by a semi-permeable membrane. On my left, I have a 20% sodium chloride solution, whereas on my right, I have 40% sodium chloride solution. Now, based on the number of sodium chlorides represented in my diagram, you can actually see that my 20% sodium chloride solution has low solute concentration, while my 40% sodium chloride solution has high solute concentration. Now, we've learned in the concept of diffusion that sodium chloride would diffuse from high solute concentration down the concentration gradient to the low solute concentration. But today's video is all about water. So let's now focus about how much water we would find in 20% sodium chloride. To make it 100%, I would need to dissolve my 20% sodium chloride in 80% water, represented by these 8 water molecules. Now my 40% sodium chloride would have to be dissolved in 60% water, represented by these 6 water molecules. Now when we compare between water molecules, you find that the 20% sodium chloride that had low solute concentration now has high water concentration, scientifically called hypoosmotic, while my 40% sodium chloride, which had high solute concentration, now has low water concentration because it only has 60% water. This we call as hyperosmotic. So what happens to water now is that it will move from a region that has high water concentration to a region that has low water concentration. Now you see we have equal amounts of water molecules. When this happens, we say the solution are isoosmotic, that is equal at 70% water. So by definition, osmosis is the movement of water from a high water concentration region to a low water concentration region across a semi-permeable membrane. Now I'm going to explain how osmosis affects living cells. Here I have two beakers, one containing hyperosmotic solution such as distilled water, and the other beaker containing hyperosmotic solution such as salt solution. Now let me place red blood cells, examples of animal cells, in each beaker. First, we'll look at the hypoosmotic solution. The distilled water in the beaker will have high water potential, while the cytoplasm will have low water potential. So what happens now is that water in the beaker will begin to diffuse into the red blood cell. Now the red blood cell has only got and outer plasma membrane. So as water begins to diffuse in, the red blood cell will begin to expand and swell. But the plasma membrane, you see, is not strong. So it will not be able to withstand the pressure caused by water. And what happens is the red blood cell will lyse. The solution now will become red due to a process known as hemolysis. Now let's have a look at the fate of the red blood cell in the hyperosmotic solution. Over here, the cytoplasm will have high water potential, while the salt solution will have low water potential. So now, water from the cell cytoplasm will begin to diffuse out of the cell, and this causes the cell to shrink. This process is known as crenation. Now let's see what happens to plant cells. Now we'll look at how osmosis affects plant cells. Here I have the same two beakers. The beaker containing distilled water to represent hyperosmotic solution, and over here a beaker containing sucrose solution to represent hyperosmotic solution. Once I place the plant cells into each beaker, we see that osmosis will occur. In the case of the hypoosmotic solution, we know that the beaker will contain high water potential, while the cytoplasm and vacuole will contain low water potential. So because of that, water from the beaker will begin to diffuse into the cell's cytoplasm and vacuole. So you can see the vacuole beginning to expand. However, the cell does not expand because it has a second outer layer made up of the rigid cellulose cell wall. So what happens is water will continuously press onto this cell wall, forming what we call as turgor pressure. So now the cell is turgid. In contrast, the cell in a hyperosmotic solution will have high water potential, while the solution itself will have low water potential. So what happens now? Water from the cytoplasm and vacuole of the cell will begin to diffuse out of the cell. When water diffuses out, the cytoplasm and vacuole will begin to shrink and pull away from the cell wall. This process is called plasmolysis. Now that you understand the concept of water potential, we can redefine osmosis as the diffusion of water from a high water potential region to a low water potential region across a semi-permeable membrane. Now, this concept of potentials can actually be represented by a symbol called psi. So in the case of water potential, we will write it as psi subscript W. And water potential, by definition, is actually the potential energy found in freely moving water. However, water potential is influenced by another two potentials. Water potential is affected by psi P, pressure potential, and psi S, solute potential, also known as osmotic potential psi O. Water, as you can see here, is moving freely due to water potential. But this free movement is affected when this water molecule is inside a plant cell, which has a rigid cell wall. Now, this water molecule will begin to experience pressure potential, which is the hydrostatic pressure exerted on water molecules in a cell. So you see, as the water tries to move, it is being pushed back in by the cell wall. So that is pressure potential. Meanwhile, inside the cell, the cytoplasm will have solutes. And what happens is the water molecules become attracted to the solutes, preventing water molecules from moving freely. This is called solute potential, the attraction force between water molecules and solute molecules that prevent water molecules from moving. So the pressure potential is usually valued positively. Solute potential is usually valued negatively. And the sum of these two potentials form the water potential, which is also valued negatively. Let me now proceed by trying to explain to you what I mean by valued positively and valued negatively. I'll begin with a beaker containing distilled water where you can see water molecules are moving freely. Firstly, you will notice that there is no cell wall. Therefore, the pressure potential of the water in this beaker will be valued at 0 kilopascals. Kilopascal is the unit for potential. Next, you notice that in distilled water, there is no solute. Therefore, the solute potential is valued at 0 kilopascals. So, if you recollect the formula for water potential, which is the sum of pressure potential and solute potential, we find that the water potential for distilled water is also 0. But let's see what happens when we add solutes. For example, let's take salt solution. Let me drop in a few salt molecules. And now we notice that since there is salt molecule present, water molecules will become attracted to the solute. So the solute potential will change. And as I said, it is valued negatively. So for example, let's say it is negative 10 kilopascals. So based on the formula for calculating water potential, the sum of pressure potential and solute potential will cause water potential now to become negative 10 kilopascals. So in a beaker, the water potential is actually equal to solute potential since pressure potential in a beaker is 0. But let's now see how these three potentials change in a cell. When I place a plant cell into distilled water, water from the beaker will diffuse into the plant cell, making it turgid. So the water molecules now will begin to push onto the cell wall, developing a pressure potential. So if we look at the formula, water potential is the sum of pressure potential and solute potential, where distilled water has 0 kilopascals of water potential. So the pressure potential now will equal to negative of the solute potential. So if say the solute potential is negative 100 kilopascals, therefore the pressure potential now will also be 100 kilopascals. In contrast, if I put a plant cell in a solution with low water potential, water will diffuse out of the plant cell into the beaker, causing the plant cell to plasmolyze. Now, since the water is not pushing onto the cell wall, we find when a cell experiences plasmolysis, the pressure potential becomes 0. When pressure potential is 0, water potential will equal to solute potential. So if the solute potential is, say, negative 100 kilopascals, then the water potential will also be negative 100 kilopascals. This concept can also be represented in the form of a graph. Now, when we place a plasmolyzed cell into distilled water, over time, the plasmolysed cell will become turgid. Now, in that duration, the water potential, the pressure potential, and the solute potential will change. The change can be recorded and plotted into a graph where the y-axis shows potentials in kilopascals and time in seconds or minutes. Now, based on our earlier discussion, we understand that when a cell is plasmolysed, the pressure potential will be 0. And the water potential will be equal to the solute potential. So we can identify these points respectively. Now, when a cell is turgid, we know that water potential will become 0 and the pressure potential will be equal to the negative of the solute potential. By joining these points together, we can develop 3 linear graphs. The graph that is positioned on the positive range will be the pressure potential graph, while the water potential and solute potential graphs will be in the negative range. It is useful for you to remember these graphs because in our STPM objective questions, they may ask you to identify them. Let's move on to apply these calculations in a question. Let's begin with a simple question. If osmotic potential of a flaccid plant cell sap is negative 1002 kilopascals, what is the water potential? So there are some clue words here. First, we start with the word flaccid. As you know, flaccid cells will have 0 kilopascals of pressure potential, meaning that the water potential will equal the solute potential. And as I mentioned earlier, osmotic potential also means solute potential. So the answer to this question will be negative 1002 kilopascals. Now let's expand the question a little bit more. Predict the water potential and direction of net movement of water in cell Q, here is cell Q, when placed beside the flaccid cell. So if we want to know direction of net movement between two cells, we need to know the water potential of each cell. We already know the water potential of the flaccid cell is negative 1002 kilopascals. We just have to solve the water potential of cell Q. Now they've given us the pressure potential as well as the solute potential. So we know water potential is the sum of these two potentials. So we add 402.1 kilopascals to negative 1213.2 kilopascals and we get negative 811.1 kilopascals. So next, we need to differentiate between the value of water potential in the flaccid cell as compared to the water potential of cell Q. Now remember, whichever value is closer to 0 will have a higher water potential. So we find since negative 811.1 is closer to 0 compared to negative 1002, we can conclude that the water potential of cell Q is higher than the flaccid cell. So this means the net movement of water will be from cell Q to the flaccid cell. So I hope this video has been helpful in understanding the concept of potential. So go on, try some past year STPM questions involving calculation to see whether you can apply this concept. That's all from me. Goodbye.